and 1084 affects a cis regulatory region of the EGA1 gene that is located 5.6 kb downstream of the EGA1 transcription unit, and that alters EGA1 transcription in such a way that the HSN neurons in hermaphrodites inappropriately die by apoptosis. What does N1084 actually affect? It affects a 9 base pair long motif, and this motif is conserved in other Cenobiditis species such as C. brixi or C. rehmannia. What does conservation mean in this context? It means that several kb downstream of the C. brixi or C. rehmannia EGA1 transcription unit, the same motif can be found. Such conservation across Cenobiditis species in general suggests functional importance, in particular when the region or motif of interest is located between transcription units and therefore in regions that are non-coding. Functional importance was furthermore suggested by the finding that the other 6 dominant EGA1 mutations identified in independent screens all affected the same small motif. Basically, all 7 dominant EGA1 mutations are G2A transitions within this 9 base pair long motif. And this motif turns out to be the binding site for a transcriptional repressor, a protein that can block transcription. This transcriptional repressor is TRA1, and TRA stands for transformer. TRA1 is a zinc finger DNA binding protein, most similar to the Gli family of transcriptional regulators in mammals. And TRA1 has a very important role during C. elegans development. TRA1 is the terminal and global regulator of somatic sexual fate. What does this mean? It means that the activity of TRA1 determines whether a worm differentiates into a hermaphrodite or a male. In embryos that carry 2 X chromosomes, TRA1 activity is high. TRA1's function is to suppress the transcription of genes required for male development and to promote the transcription of genes required for female development. For this reason, an embryo with high levels of TRA1 activity will differentiate into a hermaphrodite. Conversely, in embryos with only 1 X chromosome, TRA1 activity is low. For this reason, genes required for male development can be transcribed. Consequently, such an embryo will differentiate into a male. Why sex determination? Why is a factor involved in sex determination involved in controlling EGA1 transcription? Remember, the HSNs are sexually dimorphic. It made perfect sense. In vitro, TRA1 protein indeed can tightly bind to a DNA fragment that contains the 9 base pair long TRA1 binding site as found downstream of the EGA1 transcription unit in wild-type animals. However, it failed to bind tightly to a DNA fragment that contains the TRA1 binding site as found downstream of the EGA1 transcription unit in animals that are homozygous for any of the dominant EGA1 mutations. But remember, there are dominant and semi-dominant EGA1 mutations. When compared to its binding to a DNA fragment with a dominant mutation, TRA1 binds relatively stronger to a DNA fragment that contains a semi-dominant mutation. In other words, the ability of TRA1 to bind to the motif in vitro positively correlates with HSN survival in vivo, at least in the context of heterozygous animals. Less binding in vitro means less survival in vivo. For this reason, we put forward the following model of how TRA1 controls EGA1 transcription in the HSN neurons. In hermaphrodites, the level of TRA1 activity is high. For this reason, TRA1 binds to the TRA1 binding site downstream of the AGO1 transcription unit, thereby repressing AGO1 transcription in the HSNs. Consequently, the HSNs survive in hermaphrodites. In males, the level of TRA1 activity is too low to bind to the TRA1 binding site downstream of the AGO1 transcription unit. Consequently, AGO1 transcription is activated most likely through the action of a HSN-specific transcriptional activator of AGO1. By the way, this HSN-specific activator has so far not been identified. In the context of AGO1 transcriptional regulation, TRA1 therefore has anti-apoptotic function. Genetically speaking, it acts to block the activity of the pro-apoptotic AGO1 gene in a specific type of cell, the HSNs, and hence has anti-apoptotic activity. Now, what happens in N1084 hermaphrodites? The level of TRA1 activity is high, but because of the N1084 mutation in the TRA1 binding site, TRA1 is unable to bind well. For this reason, TRA1 is unable to repress AGO1 transcription, which results in the inappropriate activation of AGO1 transcription and the inappropriate death of the HSNs in hermaphrodites. This would also explain why the dominant AGO1 mutations are dominant in the first place. Having one of the two AGO1 loci carrying the N1084 mutation is sufficient to get inappropriate activation of AGO1 transcription from the AGO1 transcription unit in cis, and hence inappropriate HSN death. Finally, in N1084 and 3082 hermaphrodites, the AGO1 transcription unit is also inappropriately activated. However, since there is a second mutation in exon 2 in 3082, a truncated non-functional AGO1 protein is made, which is unable to activate the central apoptosis pathway. Consequently, the HSNs survive. In support of this model, an AGO1 reporter is expressed in the HSNs in males, but not hermaphrodites. However, the introduction of the N1084 mutation into the TRA1 binding site 5.6 kb downstream of the AGO1 transcription unit in this reporter results in the expression of the reporter in the HSNs of both males and hermaphrodites. In the HSN neurons, AGO1 activity appears to be regulated at the transcriptional level. Is this true for the 131 cells that normally die during hermaphrodite development as well? The 9 base pair long TRA1 binding site is specifically involved in controlling AGO1 transcription in the HSNs. We now know that there are many such conserved motifs downstream and also upstream of the AGO1 transcription unit. These motifs most likely represent binding sites for additional regulators of AGO1 transcription and hence cis-regulatory elements of the AGO1 locus. Based on genetic analyses, we suspect that AGO1 transcription is the target of 60 to 70 different transcriptional regulators that act through these cis-regulatory elements, and that each controls AGO1 transcription in a subset of the 131 cells that die during C. elegans development. The AGO1 locus therefore acts as an integrator of all the signals that control apoptosis during C. elegans development. Not only have extensive cis-regulatory elements of the AGO1 locus been identified, but also some of the transcriptional regulators or trans-acting factors of the AGO1 locus. TRA1 was the first one to be identified, and a number of investigators have since identified additional ones. Interestingly, each one of these transcriptional regulators identified to date has a homologue in humans. Not only does each one of them have a homologue in humans, but for each one of them, the human homologue has been implicated in tumorigenesis. For example, GLI, the human homologue of C. elegans TRA1, was originally found associated with glioma, which is a type of cancer that arises from glial cells. And glial cells are non-neuronal cells that provide support for neurons in the brain. And that's where glee got its name from glioma. Gliomas make up about 80% of all malignant brain tumors. Since its identification, glee has also been found transcriptionally upregulated or overexpressed in many other types of tumors. Like BCL2, glee is therefore a proto-oncogene. This is an important point. In analogy to C. elegans TRA1, the glee transcription factor may normally block the transcription of a pro-apoptotic gene. For example, one of the mammalian BH3-only genes. In tumors in which glee is upregulated or overexpressed, this would lead to the transcriptional repression of the BH3-only gene and, consequently, a block in apoptosis. This block in apoptosis could be the first step towards tumorigenesis. Transcriptional repressors of pro-apoptotic genes, such as BH3-only genes, potentially are proto-oncogenes. Proto-oncogenes are genes whose overexpression or inappropriate activation can cause tumorigenesis. BCL2 and glee are examples of such proto-oncogenes. Conversely, transcriptional activators of pro-apoptotic genes potentially are tumor suppressor genes. And tumor suppressor genes are genes whose loss can cause tumorigenesis. For example, loss of a transcriptional activator of a BH3-only gene potentially leads to the inappropriate survival of cells, which then can acquire additional defects or mutations and give rise to a tumor. An example of a transcriptional activator of AGO1 is the transcription factor SEB1. SEB1 is required to activate AGO1 transcription in germ cells in response to DNA damage. In animals lacking SEB1 function, AGO1 is not transcriptionally upregulated in response to DNA damage, which leads to the inappropriate survival of germ cells that would otherwise die. The mammalian homologue of SEB1 is p53, an important tumor suppressor that is found inactivated in more than 85% of all human cancers. One important target of p53 is the BH3-only gene PUMA, which demonstrates the importance of being able to eliminate damaged cells in response to insults, such as DNA damage. Like in the case of C. elegans AGO1, the activity of the mammalian BH3-only gene PUMA is regulated predominantly at the transcriptional level. Other mammalian BH3-only genes that are transcriptionally regulated are NOXA, HRK, and BIM.